Hello and welcome to the September and October Longevity Review. I'm combining these 2 months together in a bid to catch up with these reviews as I had to take some time off due to health reasons. In this review, we will take a look at the largest and most comprehensive dietary restriction study done in mice to date, how the rat lifespan record might have been broken by exosome treatment, how an inhibitor of hyaluronan synthesis increases mouse lifespan, how exercise intensity is more important than volume for mortality reduction, and for the Canadian content study, how a protector of neurons plays a greater role in lifespan. A reminder that this review is presented to you by the Canadian Longevity Association, Canada's premier longevity nonprofit. My name is Chris Linnell, and I am the founder and president of the association. If you'd like to find out more about what we do, then please check out the links below this video, where you will also find links to the newsletter and studies reviewed here. And lastly, don't forget to like this video and subscribe to our channel. So without further ado, let's take a look at the first study. Dietary restriction, also known as caloric restriction, is the oldest and most studied form of lifespan extension. Since it was discovered in the 1930s that if you fed rats less, they lived longer, countless studies have been done. So why include another one in the highlight section of this newsletter? Well, mainly because this quite possibly is the largest and most comprehensive study done to date. Also, they studied various forms of dietary restriction, not only reducing the calories every day, but also looking at fasting. This study was done by Calico, Google's longevity company. So they had the money to include a large number of mice. Anyway, let's take a look. Starting with the study design on the left here, there were 5 groups of mice, each consisting of 192 females. Now, they chose females because females have been understudied in longevity research up till now. They also made sure to use genetically diverse mice because in too many lifespan studies, a specific strain is used and there can be specific strain effects. However, if you use genetically diverse mice, that corresponds more closely to humans who are also genetically diverse. The first group of mice were the control mice in gray here, also denoted by AL, which stands for ad libitum, meaning they were given an all-you-could-eat buffet and could eat as much as they wanted any day of the week, as you can see by these gray squares from Monday to Sunday. Now, the first group of dietary restricted mice were fasted for 1 day, so you can see by the missing square in the middle. Now, it sort of goes over 2 days because mice are nocturnal creatures, so they normally would be eating over the course of the night. The second group in darker blue were fasting for 2 days. Again, you can see by this 2-day gap here in the middle. Then you had more traditional caloric restriction, 20% where they ate 20% less, and 40% where they ate, obviously, 40% less. Now, a nice thing about this study, which makes it very clear, is that they use the same color scheme for all of the graphs and charts, which makes it easier to follow. Now, I'm not quite sure what the sort of 3 stacked squares mean. There's no sort of explanation of that. I don't know why they couldn't have just carried this across. But anyway, yeah, those mice ate 20 and 40% less. Now, let's take a look straight away at the lifespan results. So at the top here, you've obviously got the control mice. The round circle shows the medium lifespan, or when 50% of the mice had died, and the triangle is the maximum lifespan when, generally speaking, 90% of the mice have died. So for 1 day fasting, median increased by almost 12% and maximum by just over 6%. For 2-day fasting, slightly less median increase. However, the maximum went up to almost 15%. Caloric restriction, however, did even better. So at 20%, median was 18% better than the controls, and the maximum was as far out as 22%. However, 40% had the biggest increase of all, with a 36% increase in median and a whopping 38% increase in maximum lifespan. Now let's take a look at the lifespan curves. So here on the left is, well, as I said, the lifespan curves. And once again, you can see how the control mice died first and died quickest, followed by the 2 fasting mice, single day light blue, 2-day dark blue. And you can see there's a divergence at roughly the 30-month mark, where the one-day fasted mice died off faster, and then the 20% and 40% caloric restriction with, you can see the big increase for the 40% group. Now these dotted lines are showing the median lifespan. Now on B here, this is showing the mortality doubling time, so the time taken for twice the amount of mice to die. And once again, the 40% restricted mice far outperformed the other mice. And interestingly enough, the one-day fasted mice performed slightly worse than the control mice. This can be seen on the survival curves, where there's a sort of a steeper descent for the one-day fasted mice in this portion compared to either the controls or the two-day fasted mice. And on the right here, simply this is a graph plotting the deaths of each of the individual mice. I'm assuming there are 192 dots here. I'm not counting, but you can do so if you so wish. And just to add, these boxes here, the middle line shows the median, so when 50% died, and the outer left edge shows when 25% have died, and 75% on the right-hand side. Next, they looked at body weight and composition. Starting off with body weight, you can see here that the control mice increase in body weight up to around, looks like 20 months before declining rapidly. And the same can be seen with the one-day, two-day, and 20% group. However, the 40% group, their body weight after initial climb declined quite quickly and stayed low. Now, because the mice died at very different rates, so you can see in B here, this is proportion of life lived, so you can sort of compare them. And once again, you can see that the control mice weight increased the most before declining, and the 40% decreased first and stayed stable, basically. And on the right here, we can see the impact of body weight, change in body weight, lean tissue mass, and fat tissue mass, the impact it had on lifespan at varying ages, blue meaning a positive impact, red meaning a negative impact. And the stars here show the amount of statistical significance. The more stars, the more significant it is. Now, interestingly enough, having a higher body weight at 3 months was negative. However, by 27 months, it was actually a net positive. The same can be seen with the fat tissue mass. So at 21 months and 33 months, it was beneficial to have more fat. The same can be seen in D here, where initially at 2 and 3 months and 3 and 4 months of age, mice which were heavier had a lower expected lifespan. However, this sort of disappeared as they got older. By 18 months, there was no detriment to being heavier. Now in E here, which looked at mice which were either lighter or heavier at 6 months of age in their survival curves, the mice which were lighter at 6 months of age had longer survival than at median than the heavier mice. You can see here by these dotted lines showing the median, which goes from 27 to 35 or so for the lighter mice, but is considerably lower for the heavier mice. Now, one interesting aspect is for the fasting mice, remember the blue lines, for the lighter mice, they lived longer, just like in the overall study. However, for the heavier mice, there was no actual real difference, especially at median, between themselves and the controls. There was a slight divergence then later in their lives, but at least at the medium time point, there was no difference between them and the controls. Now, F is quite interesting. So mice, when they are handled, get stressed and lose weight, and it takes a while for them to sort of recover the weight that they have lost while being handled. Now, at 10 months, and I think at 22 months, the mice were extensively handled while they were being tested. And these graphs show lifespan and how much weight they either lost or gained during that handling process. And basically, the mice which lost more weight during that handling process did not live as long, so they were less resilient. So you can see that by these upward sloping lines. So the ones on the left lost more weight, the ones on the right lost less, or even maybe gained weight. And for all of them, from the control all the way to the 40% restricted mice, there is an upward slope, especially for the 2-day fasted mice. They have the steepest gradient here. Then at the bottom here, simply showing how much lean tissue mass and fat mass changed over the course of the studies. So it looks like lean tissue mass, apart from the 40% group, increased, and fat mass decreased, especially in the 40% group. Next, they looked at health and metabolic traits. The assumption was that a healthier mouse would live longer. However, the authors found very few associations between various health metrics and lifespan. The only one that really stood out was frailty. You can see here on the top here, and also activity in the dark, which obviously are correlated. A more active mouse is unlikely to be frail. And we can see on the right here in C that frailty increased over the course of the study for all groups of mice. However, for the dietary restricted mice, the frailty increased more slowly. The importance of frailty can be seen in D here, where lifespan is on the y-axis and the amount of frailty is on the x-axis, and there was a downward slope, so that the more frail the mouse was, the shorter its lifespan. That could be seen with all groups of mice, from the control mice all the way over to the 40% restricted mice, as shown by the downward sloping lines. Least of all with the 20% restricted mice, however. They also looked at body temperature on the left and glucose on the right, but no significant associations were found. Next, they looked at the immune system and blood. First, on the left are various blood traits, again showing a correlation with lifespan, with red being negative, blue being positive. And the biggest negative correlation was the red blood cell distribution width, so how wide or large the red blood cells are, wider is worse. And number of red blood cells and hematocrit and hemoglobin had sort of positive associations. B is looking at various components of the immune system. Again, negative correlations in red, positive correlations in blue. And lymphocytes sort of stood out as having the most positive correlation. So lymphocytes are sort of immune cells which kill bacteria and stuff. And looks like memory B cells and effector CD8 plus cells had the biggest sort of negative correlation. And we can see on the right here how lymphocytes decrease with age, and the restricted mice had higher counts of lymphocytes than the control mice throughout. And the opposite is with effector CD4 plus T cells, which increase with age, and the control mice had higher amounts of those than the dietary restricted mice. And with the red blood distribution width here, that also increased with age. But in F here, we can sort of see how having a smaller red blood cell width is better. So again, lifespan on the y-axis, and on the x-axis is showing the size of the red blood cells, and the lines indicate that having bigger red blood cells is worse than having smaller ones. And the same could be seen for all groups of mice. Finally, they looked at how genetics impacted lifespan. And you can see on the left here the comparison between the dietary restriction and the genetic background of the mice, and the percentage impact they had on lifespan. And at 6 months, when there were 929 mice still alive, genetics had a much bigger impact, 23.6% compared to only 7.4% with the diet. Now, after 11 months, the genetic effects have declined to 17%, and the dietary restriction increased to 8.4%, which continued at 17 months when there are only 631 mice still alive, where the dietary component increased to 11.4% and the genetic background decreased to 15.9%. However, throughout this entire study, genes played a bigger role in the lifespan of the mice than did the dietary restriction. So if you're planning on restricting your own diet, then you better cross your fingers that you have the right genetic background. Looking more closely at a single component, they chose a haplotype, which is a cluster of genes affecting one process. And they looked at a haplotype which affects red blood distribution width. And on the right here just shows where it could be found on the chromosome 18, it looks like. And there was a certain haplotype called cast, and mice which did not have cast lived longer than the mice which did have cast. So you can look at the median lifespan of both groups. So the mice without cast had longer lifespans, I guess from 27 to just under 36, compared to looks like, I don't know, 20 to just over 30 median lifespans for all the different groups. So again, this shows the impact of genetics and how we might all be prisoners of our own genome, at least until gene therapy comes of age. So as you can see, this was a very comprehensive study, which, as usually with science, posed more questions than it answered. The biggest paradox, as the authors put it, was that mice which were lighter when they were younger lived longer, whereas mice which were heavier when they were older also lived longer, and that it seemed to be about not losing weight as you aged, which was the most important factor. So much to ponder if you're considering dietary restriction yourself. I'm very excited to bring you details of the next study, as I've been following this research group since the beginning, and the research has featured numerous times in previous newsletters. I'm also in correspondence with one of the principals, and I've got some exclusive news to share with you at the end of this section. Their work involves using a blood plasma fraction from young animals to rejuvenate older animals. But first, I want to briefly recap a study from last year to which this study is a follow-on. This study was able to reduce the epigenetic age of rats and return various biomarkers back to youthful levels. Now, the important name to notice in the list of authors is Steve Horvath, who is sort of the godfather of epigenetic clocks. And the fact that he gave his name to the study and performed the epigenetic analysis gives a lot of credence to this study. So let's quickly go through it. So here's just some epigenetic data. And down here, you can see the results of the epigenetic age tests. And briefly, on the left, the red bars are the old control animals. The blue bars are young animals, and the orange bars are the treated animals. And I should say all these studies were done in rats, and they used four different epigenetic clocks. And for each of these, looking at 4 different tissues: blood, liver, heart, and hypothalamus, which is in the brain, you can see that the orange bars are considerably lower than the red bars. And in fact, in a lot of these, closely approximating the height of the blue young animals. And all the rats started treatment at 24 months, so late middle age, earlier old age for rats. And for each of the different clocks tested, and in each of the different organs tested, the orange bars are lower than the red bars, and as I say, close to that of the blue younger animal bars. Moving swiftly on, as I only want to briefly cover this. Next, they also looked at various biomarkers from different organs, from the liver, heart, kidney, and pancreas. And you can see the days after treatment at the bottom here. And once again, the red dots are the old control animals, orange dots are the treated animals, blue dots are the young controls. And for each of these markers, you can see that over the course of 150 days, the orange dot separated from the red dots and approached that of the blue dot. So you can see that a lot of these biomarkers return to more youthful levels. Next, they did some cognitive tests where the animals had to learn how to escape from a platform by finding a hole. And once again, red is the old control animals, orange are the treated animals, and blue are the young animals. And they're tested over several days, and they improve over the course of that time. And they were tested 4 times. And at first, the treated animals had a similar escape time as the old controls, but over the course of their testing or learning experience, they started approaching that of the younger animals. And certainly by the 3rd and 4th month, or 3rd and 4th tests, they pretty much reached the same level as the younger animals, and clearly separated even at the beginning from that of the old control animals, implying that their cognition improved. Next, here are just some markers of oxidation and antioxidants. So melandialdehyde, or MDA, is a toxic substance. We want less of it. And in the brain, heart, lung, and liver, levels were much lower in the treated animals than the controls, and again, approaching the level found in the younger animals. Reduced glutathione, catalase, and superoxide dismutase are sort of three antioxidants found in our bodies, and higher levels are better. And once again, the treated animals had much higher levels than the control animals, once again approaching the levels found in the younger animals. And finally here, two markers of inflammation, interleukin-6 and tumor necrosis factor alpha. You can see that this is days after treatment. You can see that the levels of these inflammatory markers fell from that of the control animals down to levels similar to that of the young control animals, implying a lot less inflammation in their bodies. And here NRF2 is a transcription factor which helps defend our cells. And once again, the treated animals had higher levels than the old controls, approaching the levels or even exceeding the levels of the younger animals in the brain, heart, lung, and liver. Now let's take a look at the new paper. The study once again used 24-month-old rats, but this time female rats, as the previous study mainly used male rats. And they were once again given a plasma fraction from young animals. Now, one thing I didn't mention in the previous study, which made it significant and was used again in this new study, is that they didn't use the blood of young rats. They used the blood of young pigs. Now, the significance of that is that it means that this treatment crosses the species barrier. So why is that important? Well, if the same thing can work in humans, then we don't want to be getting the blood of young humans for obvious ethical reasons. If we could instead get the blood from young pigs or other animals, then there could be obviously an unlimited supply of this substance, and which bodes well for future treatments. The one thing that differentiated this study was that it was a lifespan study. So they followed the animals until death and saw how old they lived. Another difference was that they used 4 treatments in this study as opposed to 2 in the previous study. So let's take a look at the study design to start with. Now, there wasn't a nice graphical extract to show you, but I just want to mention what they did. So they were injected with this substance called E5 4 times over the course of 7 days, every second day. And then they repeated the procedure on the 68th day after the first injection, 181st, 280th, and 377th day of the study. And all the dates are from the first injections, and that's day 0. Now let's take a look at what is inside this plasma fraction. So at first, when they started experimenting a few years ago, they were just using a concentrated fraction of plasma. They weren't exactly sure what was inside it. And they since discovered or since figured out that it mainly consists of plasma-derived exosomes. So these little packets, little spherical packets of chemical information which cells use to communicate with each other, or one method they use to communicate with each other. And the first figure here simply shows these little exosomes, and this little graph here shows the size of them. They're basically between 50 and 150 nanometers. And here is just a block which shows various surface markers of these exosomes, CD9, 63, and 81, which indicates that it is indeed these exosomes. But let's look at the first results. A lot of these are similar to the previous study we briefly looked at, but first of all, we're looking at body weight. And here, the pink dots are the old controls, and the orange squares are the treated animals. And I should also mention that it was a small study. The researchers are unfortunately underfunded, so they only had 8 animals in the control and treatment groups. But what you can see here is each dot represents one of the animals' sort of weight over the course of this study in days after the very first treatment. And basically, there was no statistical difference between the 2 groups. They had the sort of same body weight increase. And this looks like there was one animal which had lower weight, and one old control animal which had higher weight, but basically there was no difference between the 2 groups. There was, however, a difference in grip strength. Once again, pink is the old control animals, orange are the old treated animals, and after 8 days, so after the first treatment had finished, there was immediate divergence, which reached statistical significance, and you can see that by these stars above these bars. The grip strength, so how strong the animals are, increased a little bit at first, but then after the second treatment, which was from 68 days, there was a marked increase. You can see the difference between the orange and the pink bars. Now, this reached statistical significance, I guess, between it looks like day 8 to day 110. I'm not sure why it wasn't statistically significant after that date, as the bars look still quite a bit higher. The treated rats were a lot stronger than the previous ones. Moving on to the markers used in the previous study, you've got biomarkers for the liver at first. So SGPT and SGOT are markers of damage to the liver primarily. You can see it starts with day 0 of the experiment and then after 420 days. At the beginning, the levels are pretty much the same between the control and the treated animals. But then after 420 days, there is a divergence, lower levels in the treated animals, which reach statistical significance. 2 dots is greater significance than 1 dot. Now, for the kidney, creatine, and bun levels, for creatine, there was no statistical difference. So the levels, just the eyeball test seems to show that they were lower here. And for bun, it did reach statistical significance. As you can see, the levels are a fair bit lower in the treated animals. And for triglycerides, which generally speaking, we want lower levels of, once again, it reached statistical significance after 420 days. Next, moving on to glutathione levels, again, one of the primary antioxidants in our bodies. And you can see here that the levels were higher in the treated animals in the brain, heart, lung, and liver. They don't mention at what time point this test was done, but anyway, the levels are considerably higher, reaching statistical significance in the heart and especially in the liver. And SOD, superoxide dismutase, once again, the levels were higher in the treated animals, reaching statistical significance in the brain, liver, and especially the lung. Finally, catalase, which reduces oxygen peroxide. It didn't reach statistical significance, but just the eyeball test shows that once again, the levels are higher in the treated animals. And MDA is a toxic substance, which we want less of. And in the treated animals, it was reduced, reaching statistical significance in the brain, liver, and especially the heart. So very good signs altogether. Next, they looked at inflammatory markers. So just like the previous study, TNF-alpha and IL-6. It's a bit bigger. So once again, pink is the old controls, orange are the treated animals from day 0 of the study until day 391, the last time they tested this. And immediately after treatment, there was a small divergence between the 2 groups of animals. And then after the second treatment, after 68 days, there was a much larger divergence, which reached statistical significance on for the days tested. So again, lower TNFA is better here. So that's a good sign. And the same goes with went with IL-6, which following treatment, it declined a little bit. And then after the second treatment, after 68 days, there was a marked divergence reaching statistical significance for a period of time. See by the asterisks of the stars there. Then it stopped. I'm not, again, sure why, as there did seem to be a difference between the 2. Anyway, next they looked at P53, which is a marker of senescence. And again, lower levels are generally better. And at day 0, they are at similar levels. And then at day 210, they had a statistical significant difference between the 2. And then you can see the levels of P53 were actually lower after 210 days than at the beginning of the treatment when the rats were 24 months old. So NFK is another sort of inflammatory marker, and once again, levels decreased significantly after 210 days compared to the control animals. So once again, all in all, this is a very good sign for this treatment. Now, these results are pretty similar to those of the previous study, but what about lifespan? So let's take a look at that. So here we have the individual lifespan of the 8 control animals and the 8 treated animals in months, with the lowest or the first to die among the control animals at 34 months and the last to die at 39.5 months. In the treated animals, the first to die was at 38 months and the last to die was at 48 months. The averages here are 37 months for the control and 40 months for the treated animals, which is a 9.7% difference. Not too shabby, but not too exciting. However, the difference in maximum lifespan between the two longest lived rodents was 21.7%. But what's interesting about the oldest lived rat in the control group, a rat named Seema, who was potentially the longest lived rat ever. In fact, she received the Diamond Longevity Award for longest living rat from the North of England Rat Society and featured in the Guardian newspaper. So, I mean, this was a very small study, only 8 rats, but still, longest lived rat ever is not a shabby thing. Now let's look at the above data in graphical form. And here you can see on the left the survival curves with the control animals dying off by 40 months and the treated animals surviving longer, with Seema ending her life way over here. And on the right here, the same data, but just indicating that at sort of 36 months, the control animals had started to die whilst all the treated animals were still alive. And by 40 months, all the control animals had died and 3 of the treated animals were still alive, with Seema getting all the way out to 48 months before succumbing. So although this is a very small study, these are very intriguing results. Now on to that exclusive news that I promised you. So I had correspondence with one of the authors, and this experimental group, or this research group, have now moved on to testing this treatment in larger animals, dogs specifically, and in fact, beagle dogs specifically. Now again, because they don't have huge amounts of funding, it's a very small study. In fact, they're only using 3 beagle dogs, one who's 9 years old and 2 which are 11 years old. And beagle dogs live on average between 12 and 15 years, but being much larger than rats, they're sort of closer to humans. And once again, they are using blood from a different species, but not pigs this time. They're actually using blood from young horses. So they're replicating the findings in a sort of different animal models, not only from pigs to rats, but now horses to dogs. And they haven't finished the study. They're hoping to publish results early next year. However, so far, the results are very encouraging. And in fact, levels of IL-6, remember that inflammatory marker, have supposedly dropped by more than 50%. And they're doing treadmill tests, and they're taking videos of the dogs. So we should actually be able to see, hopefully, some difference between pre and post treated dogs. Now, next year, they're also planning on moving into cats, specifically cats which have chronic kidney disease. Now, I wasn't told what form of blood they'll be using for the cats, but if these experiments turn out to be successful, the next step would be moving into humans. So hopefully within the next few years, fingers crossed, we might get some results in actual humans. And if they can replicate the results in rats, then it could be a real game changer. The next study is also a lifespan study, but this time in male mice, and involved an inhibitor of hyaluronan synthesis. Hyaluronan, also known as hyaluronic acid, is used in various skincare products, and also it is used in the body to lubricate joints and various other processes. And the researchers here used an inhibitor of hyaluronan synthesis for methyllumbelliferone, or 4-MU. I'm going to stick with 4-MU, thank you very much. And they used an inhibitor because although hyaluronan has beneficial effects, it also has some detrimental ones. Specifically, in older age, breakdown products or fragments of hyaluronan can be found in various inflamed tissues, and it has been implicated in insulin intolerance and the onset of diabetes. Previous research by these researchers has shown that it might be beneficial for that disease. Here, however, they tested it out for lifespan. So let's look at the results, starting with lifespan. And they gave 4-MU to the mice in their food starting at 5 weeks, which is around here in the lifespan curve. and there were 20 mice in each group. Now you can see here the control mice are in black, the 4MU treated mice are in blue, and you can clearly see a difference in the survival curves. At the median, when 50% of the mice had died, 4MU outperformed the controls by 26%, going from 122 weeks to 154 weeks. At maximum, the survival increased from 159 weeks to 194, which was a 22% increase. So quite impressive. And on the right here, you can see the hazard ratios. So this shows the chance of a mouse dying at any given time. And the 4MU mice had a 71% lower chance of dying. The researchers looked at other measures beyond lifespan, starting with glucose. And as you can see here with A, showing the glucose levels over the course of this study, with the white dots being control, the blue dots being the 4MU. At first, the treated animals had much lower levels of glucose, as you can see by these stars, which show statistical significance, before being pretty much the same for the remainder of the study. At the same time, insulin levels were also lower, as you can see by the two stars here. They reach statistical significance. And I should just mention here as well that generally speaking, and up to a certain point, lower insulin levels are better. They also tested the mice by giving them insulin and seeing how the glucose responded at 24 weeks and 130 weeks. And you can see here that the treated mice had much lower levels in response to insulin at both 24 and 130 weeks, which reached statistical significance. In terms of the sort of area under the curve, as it's called, for glucose levels, there was a difference which didn't reach statistical significance at 24 weeks, and there wasn't really any difference at all at 130 weeks. Next, they looked at insulin sensitivity. And in this experiment, which involved a different set of mice, they were basically given glucose intravenously. And the results for glucose on the left and insulin on the right. And as you can see here, there wasn't really any difference in terms of the glucose response between the control and the treated animals. However, on the right here, you can see that there was a much lower insulin spike in the treated animals compared to that of the controls, which implies that there was a much greater sensitivity to that insulin, since the glucose levels lowered at the same rate, or even maybe slightly below that, of the controls. And you can see here by the areas under the curve in C and D, so glucose, there was no real difference. However, for insulin, there was a considerable difference, which reached statistical significance. A couple other measures of insulin. Firstly, insulin sensitivity. The treated mice had greater sensitivity in this acute insulin response to glucose measurement. They had much lower levels of insulin, again, reaching statistical significance. And in the HOMA-IR index, which is a measure used in humans to diagnose diabetes, they had a much lower score on HOMA-IR. Since 4MU is an inhibitor of hyaluronan synthesis, they next wanted to see if levels of hyaluronan in the bodies of the mice decreased or not. And what they found was that although the blue bars are lower than the white bars, it did not reach statistical significance. So on the left here in A is serum, so I guess in the blood of the animals, B is muscles, C is fat, and D is the liver. And although the levels do look lower, they said long-term MU4 does not reduce serum and tissue hyaluronan, which I guess means that it doesn't interfere with the beneficial effects of hyaluronan in the body. Next, they wanted to see if these results were about the mice sort of eating less. Now, as you can see on A here on the left, which shows the weight of the mice over time, the treated mice gained a lot less weight at midlife than the control mice, which was statistically significant. But they were eating the same amount of food in grams per day as the controls, as you can sort of see here. So this was measured over the course of 4 weeks, and I guess 1 week, one mouse ate a hell of a lot. Another week, ate hardly anything. That was the same mouse, that is. Looking at cholesterol and triglyceride levels, you can see that cholesterol was slightly higher, triglycerides were slightly lower, but again, this did not reach statistical significance. So it's interesting that they could eat as much as they wanted, but didn't seem to gain any extra weight, or certainly not compared to the control mice. Looking a bit further into this, they next looked at energy expenditure. Once again, you can see here in A that the weight of the treated mice was considerably lower than the control mice, again, statistically significant. However, their food intake was the same, or roughly the same, maybe slightly lower. So this is in grams and this is food intake in calories. Slightly lower, but again, not statistically different. You can see on the D here, this shows locomotor activities, how much they sort of moved, and the treated mice actually moved maybe a little bit more. At least there were some lazy ones and some at least one very active one, though this didn't reach statistical significance. You can, however, see in the energy expenditure chart here on the bottom left, which is over the course of 124 hours, the blue lines are consistently below that of the black lines. So the treated animals expended less energy than the control mice, which can be seen in F here in bar form, which reached statistical significance, with the blue bar being quite a lot lower than the white bar. This is in calories per hour. And they also had a sort of a positive net energy balance compared to the control animals. Although this was a very small study with only 20 mice in each group, the authors state that they replicated these lifespan findings in another group of mice. And what makes this study even more intriguing from a human perspective is that 4-MU is already in human trials. Halo Bioscience is using 4-MU to treat pulmonary hypertension, and it has already passed phase 1 safety trials, meaning that it is safe in humans, and it has recently passed a phase 2A trial as well, in which it improved the 6-minute walk test and also various quality of life measures, and they're going to be moving it on to larger trials soon, hopefully. But again, this is quite promising, the fact that it has shown to be safe in humans and is progressing towards clinical application. Now, clearly a lot more research is still needed, but it's a case of watch this space. Now from research in rodents to some research in people, and research which has immediate applicability. And the research is about the amount and intensity of physical activity needed to reduce mortality. Now, this paper is quite, let's say, arcane and uses a lot of highly technical terms and is not actually the best to really go through. However, the graphical abstract is excellent, and that is what we are going to take a look at. This study used data from the National Health and Nutrition Examination Survey, a very long-running survey whereby every year several thousand Americans are questioned about their nutrition and various measurements are taken. And a lot of observational trials use data from this survey. And in this particular study, they used participants from the 2011 to 2014 intake, and there were roughly 7,500 of them, all over the age of 20, and they gave them activity monitors, which they wore for 7 days to measure their physical activity over the course of that week. And they were more sophisticated than simple step counters, showing the intensity of the exercise as well. And they were followed for almost 7 years, looking at all-cause and cardiovascular disease mortality. And what they found, unsurprisingly, was that exercise is good for you. So, of course, you knew this already. However, they went into a little bit more detail, and one thing they found was that the intensity distribution was more important than the volume of exercise. So intensity distribution is just how intense you're exercising, from walking to running, for example. And you can see here on the left, which shows the intensity. And again, the hazard ratio shows the likelihood of dying, and lower is basically better. And across the x-axis at the bottom, it shows how intense the exercise is. And there was a sharp decline in the hazard ratio the more intense the exercise was. And at the bottom here, you can sort of see the number of people performing the exercise at the various intensities. Less intense on the left, more intense on the right. The middle line shows, I guess, the median, the middle of the pack. The two other lines show the 25% and the 75% values. And so below this dotted line shows better than average. But you can see by the shape of this red curve, it although goes below it for a while, it starts curving up again. So there's the indication that too much intense exercise is also not necessarily good for you. And on the right here, it's showing the similar thing, but for volume. Once again, the hazard ratio and the slope here going to the halfway or median mark to the right is better. But again, it starts curving upwards, again, indicating that perhaps too much exercise in terms of volume is not necessarily the best. Like, don't run a marathon every day. Again, you can have too much of a good thing for anything. But again, according to the researchers, intensity was more important than volume for reducing mortality risk. The next thing they looked at was how fragmented the exercise was, or what the duration was. because recently there's been a lot of talk in the WHO, for example, saying that, any little bit of exercise is beneficial, which is correct. 30 seconds of walking or running is better than 30 seconds of sitting on your ass. However, putting those 30 seconds of activity together as opposed to separating out is better yet. As you can see here, they give the example of a 30-second moderate run. So if in a day you ran for 30 seconds 10 times, equaling 5 minutes, that wouldn't be as good as if you'd run for 5 minutes in a single instance. So if you do get around to going on that run, then make sure that it's for at least 5 minutes. And finally, the Canadian content study, which comes from McMaster University and the lab of Bhagwati Gupta. They looked at a protein called mesencephalic astrocyte-derived neurotrophic factor, or MANF, which has been known to support neuronal growth, survival, and differentiation. But they have found that it plays a greater role in cell survival as well as organism lifespan. Before we get into it, I should say that this paper is unfortunately behind a paywall. However, there is a press release you can take a look at, and fortunately, the paper also exists as a preprint, and it is the preprint we are going to take a look at. Now, I've had correspondence with Bhagwati, and he sent me the final paper, and I sort of compared the preprint with the final paper, and although there's some text differences and some of the figures have been moved around, it is fundamentally the same paper, and none of the major findings have changed. So let's take a look at the preprint. In this paper, the researchers used C. elegans worms, which are one of the basic model organisms used in biological research. And they started off by creating transgenic worms, which lack the ability to produce MANF. So let's see what that resulted in. And first, they gave these worms a stressor called tonikamycin, causes the worms to stress out. And in the middle here actually are some normal worms. On the left are the unstressed worms, and on the right in blue are the worms with added toni, and the levels of stress increased. Now here on the left are the mutant worms on day 1 as well, and the levels of stress increased a lot more when they were given toni, and this continued on day 3 adults, as you can see here. Now they created 2 mutant strains that lacked MANF, and here in F and G you can see the other strain, which also saw a lot more stress when they were given this stressor. And when they checked the lifespan of the worms, you can see an I and J here on the left once this text goes away. On I on the left is just sort of the worms without the stressor, and N2 the sort of the pink line are the normal worms, and the 2 mutant worms in different shades of blue. And you can see that their lifespan was a little bit less than the normal worms under no stress. And then in J here, you can see when one of the mutant worms was given the stressor, they had a larger decrease in lifespan. And K here shows when they were given an acute dose of tonisamycin, again not living as long as the normal worms, with the mutant worms being the blue column. Next, they looked at protein aggregation or protein clumping in the mutant worms. And they looked at 2 proteins, acyclovir and polyQ. And acyclovir is implicated in diseases such as Parkinson's, where its clumping in the brain causes neurons to die. And what they found was that there was a lot more clumping in the mutant worms. So here, for example, the purple is the normal worms, blue bars are the mutant worms, and the bars are basically higher after day 1, day 4, and day 7. And the same goes with this polyQ. If you look here in G and H, and also in I, it shows how much the worms were moving, and the mutant worms with more polyQs had less movement. Then they asked what would happen if the worms had more MANF as opposed to no MANF. So they created some transgenic worms which produced more MANF. And first they checked the levels of MANF in normal conditions and also when they were subjected to 31 degrees heat. And the first column are the normal worms. Next one is the mutant worms. Same goes here. The first column is the normal worms. Second one are the mutant worms. And under normal conditions here on the left, the mutant worms had a little bit more MANF expression, but under these stressful conditions, the MANF expression was quite a bit more. And this was for 1-day adults. And in normal conditions, after day 3 and day 7, you can see that the levels of MANF were also higher in the worms expressing more of it. And on the right here, showing neuronal damage with, I believe, 1 being less, 4 being more. And the normal control worms had more damage, basically, in their neurons. They looked at clumping as well in 4-day adults. If you look at D here, under 31-degree heat stress, and the mutant worms on the right had a lot less of this clumping of alpha-synuclein. Finally, they looked at the survival curves for these mutant worms, and the black line are the normal worms, the purpley pink are the extra MANF worms, and they also looked at them when they were subjected to the heat stress, and the turquoise are the normal ones under the heat stress, and the light purple are the extra MANF ones under the heat stress. You can see that the mutant worms with extra MANF lived longer both in normal conditions and under 31-degree heat stress. Under normal conditions, they lived 23% longer, and under the heat stress conditions, they lived on average 5% longer. Now, I should mention the one only difference I saw between this preprint and the final version of the paper was the figure for survival. And in the preprint, it says they lived 21% longer under normal conditions. And in the final paper, it was 23%. So there's a small change there. But nonetheless, it's very interesting to see that MANF increased the worm lifespan. So those were the results in one strain overexpressing MANF, and they did the same with another mutant strain overexpressing MANF. And they had very similar results. Here in C, you can see that neuronal damage was less. The bar on the right is the overexpressing worm, so they have less of 4, more of 1, so 1 being less damage, 4 being more damage. They had less protein clumping of A-synuclein. Blue on the right is the overexpressing worms. Poly-Q also was decreased. You can see in G and H. On the right, it's slightly less, and the worm movement increased compared to controls. You can see once this goes away, on the right-hand side, their movement was greater than that of the normal worms. And then on the bottom left in K and L here, once again, we have the worms in normal conditions, and the MANF overexpressing worms lived 19.2% longer as a median compared to the normal worms. And on the right in L there is when the worms were given that stressor, tunicimycin, and once again, the mutant worms with extra MANF lived longer than the normal worms. Now, those were the main points I wanted to share with you in this paper, but we will also briefly look at what else they found out. Firstly, they looked at where MANF could be found within the C. elegans worms, and here you can see the worms in question. They're really quite tiny, and basically they found that they were spread throughout most of the worm, mainly concentrated in the lysosomes. So the lysosomes are the sort of recycling centers. They break proteins down so that they can be used to make new proteins and are central to the autophagy process. Again, autophagy is a recycling mechanism within cells. Next, they looked at the differences in genes and transcription between the different mutants, and won't sort of go into that, but it once again looked like it was mainly occurring in the lysosome. And lastly, they looked at this process in more detail, and they found that MANF works its magic via another protein called HLH30, which is a master regulator of the lysosome and autophagy. So these are some very interesting results. Now, this is very basic science, again, working in the most simple model organism. However, MANF exists in all of us, and if they can figure out exactly how it works in the worms and how it can increase their survival, maybe in the future that could be applied to us. Now, I want to finish off couple things. Firstly, just by reading the final paragraph here for you all. So our results have broad implications for understanding the function of MANF in higher eukaryotes. So eukaryotes are organisms which have a nucleus with a membrane, and that includes us, all animals, all mammals, and manipulating its role in promoting healthy aging and treat diseases, including lysosomal disorders. In this regard, it is worth noting that MANF expression is down-regulated in Niemann-Pick and Gaucher disease models. Future work will help to understand the precise mechanisms of MANF localization, HLH30/TFEB interaction, and stress response signaling that collectively promote neuronal health, proteostasis maintenance, so that keeping the proteins in balance, and, my favorite word, longevity. And to finish off, I just want to read the correspondence I received from Sprague Dawley about their current and future work. And he says that their current research explores the proteins that interact with MANF to mediate its function and how secreted MANF affects different tissues and neuronal cells. We are also investigating the dynamics of MANF expression to better understand its role in cellular processes. So again, if they can figure out more how MANF works, that means we have a greater chance of being able to manipulate it in us and hopefully give our cells greater stress resistance, and thus as an organism, give us longer and healthier lifespans. And that brings us to the end of this review. But before you go, please don't forget to like this video and subscribe to our channel. You can also check out our newly opened merchandise store via a link below this video. So until next month, goodbye.